marine biology, and we talked about how we got to where we are today and how we got to marine laboratories. And then we ended yesterday with talking about like the main difference between marine biologists back in the day and present day. Right? And that main difference was the scientific method, right? Okay. So naturalists, marine biologists back in the day were considered to be naturalists, where they just made observations, they just wrote stuff down. And today we use scientific method to do experiments and test things. So the scientific method is a system that we use to learn about the world, and we run experiments based on questions that we have that we get from observing nature. All right? So these are the general steps of a scientific method. Really, each scientist kind of has their own way of doing things, but these are the general steps that are pretty much the same throughout. Right? They may call them different things, but it's pretty much the same. Okay? So our first step that we do is observation. Okay? So observation is the first thing. And basically what an observation is you're using all of your senses to notice things about the world around you. So you smell something, you see something, you hear something. Typically it's not taste. You don't like to use sense of taste in your science because that can be bad. But you use your senses to notice things about you, around you. When you make an observation, it's not just like, oh, the sky is blue, okay? In science, you actually are kind of specific when you make an observation. You need to include certain things when you write that observation down. There's 4 things that you actually have to include. It's 4 W's. Where, when, whether, and what, okay? So where, when, whether, what, that's what you have to include, okay? So the reason why you have to include these things is you need to be able to make comparisons between observations. So let's say you go to Malibu, to the beach, and you go today after school, right? So August 21st, you're going to go to school or to the beach on August 21st in Malibu, and it's sunny outside, okay? And you notice that in the intertidal zone, you've got a bunch of limpets that are all like spread out, okay? Let's say you go back in December, okay, and it's cloudy, and it's that in the evening, okay, and you notice that the limpets are all clumped together. So you need to record all of these things, okay, to make sure that you can go back and like compare the 2 observations and be like, oh, I wonder if the weather is what is affecting the behavior of the limpets. Okay, does that make sense? Yes, no, maybe so. Okay, so you need to record where, when, whether, and what to be able to make comparisons. So on the next slide, I have 3 pictures, okay, and there's a slot in your notes, and I want you to just write down 3 things that you notice about these pictures.
I'm going to have to use all of the observation methods.
No, just 3 things that you notice. You don't need to do where, when, whether, what. Okay, so these are mussels, okay? This is like where the water is in this picture. This is water here, those are mussels, and then the water would be down here on the bottom for this one. Here are some things that you could have noticed about those pictures. The starfish are found near or among the mussels. So in the pictures, you've got mussels and the starfish are found close to them, right? Or like actually in and among them, okay? You could have noticed that the starfish are closer to the water than the mussels are, right? So mussels seem to be up higher, like Joseph said, okay? And then in this picture right here, you've got like open mussel shells around the sea stars. It might be kind of hard for you to see that, but you've got open mussels around there, okay? As you go farther north on the coast, you get more and more starfish, right? So up here in British Columbia, like there's tons and tons of starfish, okay? And then the mussels cover large areas in the intertidal zone. So those are just some examples of stuff you could have noticed, all right? So after we've made an observation, we are now going to use a couple types of logic to come up with our hypothesis and our prediction, okay? And in order to come up with our hypothesis and prediction, the first thing that we have to do is use inductive reasoning, okay, to come up with a general explanation of what we just observed. So inductive reasoning goes from something specific to something general. Okay? So for us, let's say that our specific observation was that the starfish are found near or among the mussels. Okay, so that was our specific thing that we observed. Inductive reasoning takes that specific thing and you come up with a general explanation for it. All right, so we would say, okay, our general explanation would be that the starfish are found near and among the mussels because the starfish are eating the mussels. Right? That's our explanation for why that's happening. Does that make sense? Okay, so after you've got your general explanation, okay, you're going to use that general explanation to come up with a hypothesis and a prediction using deductive reasoning. Okay? The definition of your hypothesis is a testable educated explanation for that general explanation that you developed using your inductive reasoning. Okay? So our general explanation was that the starfish are eating the mussels. So we're going to come up with a hypothesis, a testable educated explanation for that general explanation. Okay? So deductive reasoning takes a general explanation and forms a specific testable prediction. So our general explanation was that the starfish are eating mussels. Deductive reasoning would take that and say, okay, so if the starfish are eating the mussels, then I predict that the number of live mussels will decrease over time if a starfish is present. Does that make sense? You took that big general explanation and you made it into something very specific and testable. Yeah? Okay. It would also say if no sea star is present, then the number of live mussels will stay the same. Or it should, right? So you have a hypothesis and a prediction. Now you get to set up your experiment to test that hypothesis. When you set up an experiment, you do what's called a controlled experiment. And in a controlled experiment, you have two groups. You have a control group and an experimental group. Okay? The control group is the group that you leave alone, and it acts as a reference. And the experimental group is the one that you change something in to actually test your hypothesis. Okay? So got a couple little pictures here to represent our control group and our experimental group. So our hypothesis was that if you have mussels with a sea star, the number of mussels will decrease over time, right? Live mussels will decrease. So that's what we're testing, whether or not the sea stars eat the mussels. So we're going to set up two groups, the control group and the experimental group. The little circles represent the mussels, right, in both groups, and then the star represents the sea star. Okay. So when you set up a controlled experiment, you've got things that will affect the outcome of your experiment. And anything that can affect the outcome of your experiment is called a variable. Okay? It's something that can change your outcome. We have a couple different kinds of variables when we set up an experiment. We have what are called controlled variables, and we have what are called your testable or manipulated variables. The controlled variables are things that you keep the same between the control and the experimental group. Okay, so they remain the same. I'm going to go back to the last slide, but I'll come back to this one. Okay, so when you set up this experiment with your control group here on the left and your experimental group on the right, you're going to set both of these things up exactly the same way. Okay? And the controlled variables are the things that you keep the same between both of these groups. So if you notice, we have 5 mussels in the control group and 5 mussels in the experimental group, right? What else do you think you might need to keep the same between these two things that might affect the outcome of this experiment?
Sea star.
Okay. The sea star, the sea star is actually your manipulated variable. That's the one thing that's different between the two groups. But what are something that you would want to keep the same?
Mussels.
Okay, the muscles, what about the muscles?
Size.
Their size, good. What else?
Number.
The number of muscles,
Location.
the location, good. The amount of water, the depth of the water, right? Anything else? Salinity, pH, okay? The time you run the experiment, all that stuff needs to be the same, right? So, because if you keep those things the same between these two groups, that means when you get your results, okay, you know, let's say there's more dead muscles in the experimental group than the control group. If you've kept all of those variables the same except for that C star, then you know that your results are because the C star is present, not because of something else that you forgot to control that could affect your outcome. Does that make sense? Okay, so your controlled variables, things you keep the same. The testable variable, or manipulated variable, that's the one thing that's different. For us, that is the presence or the absence of the C star. Okay, and the experimental group is the group that actually has that one thing that's different. So in the previous slide, the tank on the right has the C star in it, and so that's our experimental group. So testable variable, the one thing that's different. You want everything the same except for one thing. Okay. So here's our experiment again. Okay, and then here's just some examples of some of the types of controlled variables. And then your testable variable is the presence of the C star. So which one of these, the right or the left, is the control group?
All right.
Good job. Okay, and which one's the experimental group?
Left.
The left. Good. So you're going to see questions like this on your test, where I'll give you a scenario and I'll say, okay, what are the controlled variables? What's the testable variable? What's the control group? What's the experimental group? Okay? You're going to do a worksheet in class to help you practice and get used to doing that. All right? So if you don't know how to do it, get some help before the test. Okay, so we've set up our experiment and we run our experiment. Okay? It is not enough in science to run an experiment just once. Okay? You never know. Like if you run an experiment only one time, there could be some variable that you forget to control and it could give you results that are actually not true. Okay? So in science, you actually have to replicate your experiment at least 3 times for you to have any sort of backing to say, okay, these were my results. So at least 3 times. And it has to be both the control and the experimental group 3 times. All right? So run it 3 times. Then, in order for your results to have any sort of validity as well, you need to be able to have your experiment be repeatable. So I need to be able to take my experimental procedures and give them to somebody else, things to set it up like I did, and then get the same result. So it has to be replicated and repeatable. Okay. So these represent our 3 replicates. We're going to set it up 3 times, do it 3 times. Okay? Okay, so while you run your experiments, you're going to be collecting what's called data. Okay? And that data is the information that you collect that will either prove or disprove your hypothesis. So for us, the data that we would want to collect to prove or disprove our hypothesis would be the number of muscles that die in each group. Right? So that's the information that we need to collect. So we would probably collect that in some sort of table like this. So our results, let's say trial number 1, just remember you run it 3 times. In the control group, you get 2 dead muscles, and then with the C star, you get 6. Trial number 2, 1 dead in control, 5 in experimental. Number 3, 3 dead in the control, 7 in the experimental. All right? So that's the data that you collect. After that, you're going to go into your results. And your results are when you take that data and you analyze it and you put it in some sort of table or graph form. Okay, so for us, Okay, we're going to take and we're going to average these numbers together. So we're going to get an average of 2 dead in the control group and then 6 dead in the C-star group, or the experimental group. And we'll put that in a graph form here on the right. And so you can see average of 2 in the control and then 6 in the experimental. Yeah, makes sense? Data collection and results. Once you do that, okay, you're going to come to a conclusion to whether or not your hypothesis was correct. So, our hypothesis that the number of live muscles will decrease over time if the C-star is present, do our results support that or disprove that? We have an average of 2 dead in the control and 6 dead in the experimental. Was it supported or disproven? Supports it, right? So you can say, okay, my data shows that my hypothesis was correct, that the C-stars are actually eating the mussels. Right? So that you come to some sort of conclusion. The cool thing about the scientific method is that even if your hypothesis was incorrect, you still learned something. So you learned that the C-stars are found in the mussels, not because the mussels are being eaten by the C-stars, but something else is going on. Right? So you still learned something, which is awesome. So, when we come to conclusions, those typically lead to new questions and new observations, which leads to a new hypothesis and a new experiment. Okay, so for us, if our hypothesis was supported, we'd say, okay, it was supported, so now I wonder what size mussels do C-stars prefer? Is there a certain size that they like? I don't know. Let's see. And if no, right, so the C-stars aren't eating the mussels, so I guess, you know, I saw that there were mussels up high and then the C-stars were down low. I wonder if it's like the sharp edges of the mussels that are keeping the C-stars from moving up, right? So always comes up, you always come up with new questions. Okay. If your hypothesis was supported and, you know, the experimental procedure gets passed around the world and everybody does your experiment and you always get your hypothesis supported, that hypothesis can become a theory. Okay? And a theory is a hypothesis that has been tested over and over and over again and has not been disproven. Or a theory can always be disproven. Okay? So if you can find an experiment that is replicatable and repeatable that disproves a theory, you can disprove a theory. So like the theory of gravity, right? It says that like every time I drop this little shark, it's going to fall to the ground, right? If you can find an experiment that's replicated and repeatable where when you drop this little shark, it like floats up into the air, okay, then, and you're not in outer space, right, then you could disprove the theory of gravity. Good luck, have fun, but technically you could disprove the theory of gravity. I'm not cheating by using a string or something. An actual scientific experiment, okay? Some sample theories that we'll talk about. We'll talk about plate tectonics, how the surface of the Earth is broken into different plates. Evolution is a theory. It gets presented as fact a lot of times, but it's a theory. Creation is also a theory. Okay, so those are just some sample theories. Okay, so here's a little summary chart of your scientific method. So observations, first step. Okay, use inductive reasoning to come up with general explanation. Then use deductive reasoning to come up with a hypothesis, run your controlled experiment, collect data, get results, form a conclusion, which leads to new observations and questions, and the cycle continues. Yeah? Okay. Sometimes it is not possible to run an experiment on certain things. Like, good luck getting a blue whale into a laboratory to run an experiment. Those things are huge. So it's not possible to run experiments on them. Or let's say the animal that you're studying is endangered, and so there's no way that the government's going to allow you to do any sort of experiments on them. In cases like that, we're going to use something different. It's called directed observation, where we basically take a question that we have, and we make observations specific to that question. So, let me scroll this down. Okay. Let's say that we notice that at a certain time of year, in this one spot, there's a bunch of sharks. Okay, and so we wonder, are these the same sharks that are coming here every time this year? Or, sorry, at the same time every year, or are they different sharks that show up? So we're going to do directed observation to figure out if that's what's going on. Right? So we may do stuff like tag the sharks and see where they go and if they come back to the same spot. We may do, like, take genetic samples, so take little pieces like their skin and then compare the DNA to see if they're the same animals. Do all sorts of things, photo IDs, okay? But that's basically, we have a question and we make observations based on that question. Because you can't run a controlled experiment. Does that make sense? Okay. Next slide is a Save the Ocean action. So we'll do these throughout the year, a few throughout the year. Just some ways to help you conserve our oceans. And one of the things that you could do is use reusable mugs, okay, or reusable water bottles. Good. When you use, like, I have a horrible example of this today actually, like plastic like this, when it gets into the ocean, plastic needs to be heated up to break down, and water cools it, so it actually will take over 500 years for like a Starbucks lid to break down in the ocean. So once it gets in there, it's there for 500 years, causing problems. So use a reusable mug, recycle stuff so that it doesn't get into the ocean and cause big problems. Yeah?